Hi, we're going to be doing a lesson today on inheritance. This includes Gregor Mendel and the discovery that he made with pea plants. Okay, so the standard that we'll be in today is using Mendel's law of segregation and independent assortment to analyze patterns of inheritance. Okay, so yesterday you guys did a reading, but today, so we have Gregor Mendel. He was an Austrian scientist and priest that began modern science of genetics. He was in charge of monastery garden and used peas to create an experiment. So he actually cross-pollinated, which we'll talk about in a minute, 2 peas, and that's what the picture in the background shows you. So let's talk a little more about Mendel's experiment. So here's like a step-by-step, kind of very short compared to yesterday's reading. Pea flowers normally self-pollinate, which means they have a sperm and an egg in the same plant. So if you look at this picture here, the anther is the male part and the filament is, or the stigma is the female. So the stamen, so this is all male, this is all female, and usually like it'll pollinate itself. But what he did was he removed the anthers. And then he would like pollinate the 2 traits that he wanted. So if he had 1 pink plant or 1 or purple plant and 1 white plant, he would cross those 2. He crossed his stocks of true breeding plants. This gave him complete control over the traits that he was breeding. The cross-pollinating allowed Mendel to cross plants with different traits. So he basically made a controlled experiment. As you know, experiments are so important that they're controlled. And in order to make it controlled, he took out the variable of having both male and female parts on 1 plant. So genes and alleles. So the original pair of plants are always P for parental. So this will be your P generation. So this will be the first generation of plants to be crossed. So in this case, you have 1 purple and 1 white flower. Their offspring is called F1 generation. The F stands for filial. Filial is the Latin word for son or daughter. That's what the F comes from, filial. So parental, and then you actually can see in the experiment, after he crossed those 2, he got 4 purple plants. So what are the difference between genes and alleles? So your gene, if you look at this picture here, so these 2 are recognizing your genes, are individual characteristics that are determined by factors that are passed from 1 generation to the next. So it'd be like a certain thing. So it'd be like eye color, so or hair color. So more like the category. The gene would be like that. Allele would be the different form of genes. So tall versus short, brown hair versus blonde hair, or in this case, brown, blue, emerald, or gray eyes. If you did, so the gene would be the hair color, and the allele would be blonde, red, brown, or black. So your gene is more of the category, and then your allele would be the specific type to it. Okay, so principle of dominance. This is where we get dominant versus recessive traits. Some alleles are dominant and some are recessive. A dominant trait is going to be stronger, and it is always going to be, it's always going to be shown with a capital letter. Your recessive trait is weaker and will always be overpowered by the dominant trait unless it's homozygous, which we'll get to. And that will be represented by a little letter. So dominant is capital, recessive is little. If an organism has both dominant and recessive alleles for a trait, then they will exhibit dominant characteristics. So the purple plant was dominant, the white plant was recessive, and when you bred them, so this is the P, parental, this is your F1, and when you breed them, you saw that all that was there was left, or what you saw was the dominant characteristic. Probability and heredity. Probability, so during gamete formation, so when you make eggs and sperm, and guys, think back to meiosis. During meiosis, it's very random, right? We have crossing over, and due to crossing over, it makes it random what alleles are where. And it's almost as random as a coin flip. Homozygous means the organism has 2 identical alleles for a particular gene. So either it's going to be homozygous dominant, that's this one right here, or homozygous recessive, and this should be 2 little g's. I'll fix this on your guided notes. Okay, so imagine this being 2 little g's. I think when I press enter, it probably automatically capitalized it. So 2 it has to be either 2 capital or 2, homo means the same, right? Homo, 2 capital or 2 lowercase. The only time you can see a recessive trait is if you have 2 it has to be homozygous. Heterozygous are an organism with two different alleles, so one capital, one lowercase. The dominant allele will overpower the recessive. So even though you have the recessive trait here, so let's say this was for like green versus white, the capital G would over-dominate your lowercase. And here's just a picture to show. So you have homozygous, either two dominant, so homozygous dominant traits, or homozygous recessive, which is two lowercase. Then you have heterozygous, which is one capital, one lower, one capital, one lower. Okay, so genotypes versus phenotypes. Genotypes, genes, phenotypes, physical. Genotypes are your genetic makeup, and your phenotypes are your physical traits. So in this case, your genotype would be the fact that the chromosome or whatever gene that you were passed on, that would be in your genes, during your DNA. And the phenotypes, your physical traits, would be like purple versus white flowers. Punnett squares. So Punnett squares are a way to use mathematical probability to predict genotype and phenotype combination. I am not going to show you guys how to do this here. We will learn how to do that on Friday. So you have, these are, in this case here, hopefully we can look at this and tell it's heterozygous and heterozygous. So you have two heterozygous purple plants. That means it carries the trait for a white plant, but because it's heterozygous, it shows that it's purple. And when you cross them, you're going to get 3 purple plants and 1 white recessive, because the only time you have a recessive trait if it is homozygous recessive. It has to be homozygous. Okay, independent assortment. This has to do with meiosis. The genes for different traits can segregate independently during the formation of gametes. So during meiosis, when you go from 1 cell to 4, the splitting up, they're going to split up from each other. Okay, and when they do that, when they make their 4 cells, that is what's called independent assortment. Incomplete dominance is when some alleles are not dominant or recessive, they mix. So if you ever have roses are a really great example. If you have a red rose and a white rose, you can make a pink rose. So instead of being red or white, it's incomplete. Therefore, you get like a mix. And the last one is codominance. Both alleles are expressed in the phenotype. 2 examples here. The first one is when you have black feathers and white feathers, create speckled black and white feathers. Also, you can have a red cow and a white cow, and it'll give you a white speckled cow. Okay, that's it. Very vocabulary based, so we will have a little vocab thing after this, but that's it.